Okay, so I'm gonna really cruise through the nervous system, I'm sorry, the lymphatic system stuff. And skip a lot of stuff that you don't need to know. You guys, if you're going on to pathophys, if you're going on to microbio, you're gonna get it in a lot of detail. If you've already been to microbio, you probably know a lot of this. But I'm gonna kind of just touch on what you need to know. If you remember last time we talked a little bit about the various cell types, we talked about basophils, neutrophils. What's the most abundant one in the blood? Neutrophils. All right. Tumor specific agents, or I'm sorry, antigens. These are your natural killer cells. If you remember, I was saying they're kind of going around and they're looking at cells that are natural to the body that may have some abnormalities to them. When they have an abnormality, they may project a specific antigen that basically is signaling, hey, something's wrong with us. And those natural killer cells are going to pick that up. So what is that? It is called the tumor-specific antigen. So you should be aware of that. I'm probably going to ask you a question about that, so you want to make sure you know that one. Some cancer cells kind of sneak through and they don't get that tumor antigen, tumor-specific antigen. So the natural killer cells don't take care of them, and then we see cancer kind of pop up or metastasize and move around in the body, which can cause obviously big problems. Cells that have viruses will also so basically we say it's monitoring natural cells, but what you'll see is sometimes there are natural cells that are infected with stuff that doesn't belong in the body. So viruses, usually a virus goes inside of a cell, and then that cell will put parts of that virus out on its surface, and now the natural killer cells will go and attack that cell that belongs in the body, but it does it because it's presenting that it's infected with something. All right. See, they do. They're just sneaky, or they don't get the antigen for whatever reason, that tumor antigen, or they miss screening through the screening process. Somehow they slip through with another cell, and then they don't get picked up, which is why we can see cancer move. And it's unfortunately, much more common than you realize, probably. All right, interferons. I always say that interferons are like little cries for help. They basically alert other cells in a close proximity that, hey, we have some sort of a virus infection. I've been attacked by a virus. So interferons basically will cause other tissues to secrete this antiviral protein to protect them. Some of our interferons will secrete those chemicals, which will cause macrophages and things to migrate to those areas. Do you remember when we follow the chemical signal? What did we call that? Pretty sure I talked about that. That calls all the immune cells to the area. Yes, chemotaxis. So interferons are one of those signals that will create kind of that chemotaxis signal. Oh my gosh, my little thing is not working. Sorry. You do not have to know the types of interferons for this test. Just know in general what interferons are. This shows some of your interferons. You can kind of look through that because it's interesting. I'm going to skip a lot of these. You should know inflammation. What is inflammation? It's redness, swelling, heat, and pain. What are the cells that cause inflammation or kind of send out the signal for inflammation? Do you guys remember? Mast cells. What do mast cells secrete? Histamine and heparin. And these guys are my inflammatory response. So with histamines and heparin come all of these: redness, swelling, heat, and pain. Well, the pain, some of the pain comes from swelling, some of the pain comes from damaged tissue or whatever. infection. Yep, so mast cells are your cells that do histamine and heparin. You'll also see some of your basophils do that as well. Mast cells, basophils.
Those cause inflammation or?
Yeah, histamine and heparin cause an inflammatory response. Why? Because what we do is we basically thin the blood, we make capillaries more permeable to let things come out of it. So by doing that and increasing blood supply to the area, now we can bring all this stuff to the area, immune cells, macrophages, neutrophils, microphages, stuff that's going to attack foreign bacteria, that is going to eat up necrotic tissue. It's also going to bring repair cells. But we basically flood this area with cells that are going to help to beat infection, get rid of damaged tissues, and then repair the area. But in that process, we get these guys right here, redness, swelling, heat, and pain, which we promptly try to get rid of, right? It's actually a good thing for our body, but we hate it. So we ice it and we elevate it. We do all those things. All right, so effects of inflammation. We just kind of talked about this. It is basically taking care of pathogens and preventing or temporarily repairing injuries. And then eventually we're going to facilitate the regeneration. So you should definitely know this. Is everybody good? Necrosis is local tissue destruction. Oftentimes we think of necrosis in a bad way because like gangrene is necrosis, necrotic tissue. But really, anytime you have damaged tissue, you're going to have some amount of necrosis, which is just breaking down and getting rid of that damaged tissue. Pus is basically a mixture of debris, fluid, dead and dying cells, and necrotic tissues. One of the components to pus is dead neutrophils. Neutrophils are kind of those first guys that show up on the scene besides the localized macrophages. And I would say that a lot of neutrophils die in the battle. And so their poor little dead cell bodies are there and are oftentimes part of the pus as well. An abscess is just an accumulation of pus under the skin. Is everybody good? Anybody want more time? That's all right. All right, so this walks you through it. Here we have tissue damage. We're going to cause an inflammatory response. Who does that? My mast cells. They release histamine and heparin. These are going to open up the blood vessels, thin our blood. It causes blood flow, high blood flow to the area, which causes my swelling, heat. We'll get clot formation. We're going to attract phagocytes, especially neutrophils. Chemotaxis is one of the ways we'll attract to them. They're going to break down any damaged tissue, but also any foreign things. They can activate specific defense. What was specific defense? Yeah, it knows exactly what it's after, right? What was innate defense? It's what? Someone mumbled. Innate are things you don't have to work to get. You're born with them. So they're things like it can be your skin stopping stuff, but we can also have immunities that we're born with from our mother, maternal stuff. All right, and then we get our tissue repair. Fever, what's so great about the fever? It's a pain, but it's actually keeping proteins from multiplying from pathogens. So it's slowing replication of bad pathogens. So it's inhibiting those viruses and bacteria that are those bad foreign pathogens. It's also increasing metabolic rate, which is going to help to increase our repair, our breakdown and our repair. But probably the biggest thing it does is really inhibit protein replication because the temperatures are too high. We denature proteins at high temperatures, right? So if we turn the heat up here, we can't see that protein replication, which we desperately need to make new cells for mitosis, meiosis type things. Was everybody good? Pyrogens are just fever-inducing agents. And here we see some things that cause fevers. Interleukin, what was a interleukin? Interleukin and interferons are both kind of chemical signals. So chemical signals that say, hey, we got something going right here. And then tumor necrosis factor is the breaking down of those tumors when we've identified a cell is a tumor. So these guys turn the heat up. All right, adaptive or specific defenses. This is when we kind of fire up our B cells and our T cells. Why do we call it a T cell? It comes from the thymus. It matures in the thymus. The thymus has a blood, remember how we had a blood-brain barrier? We have a blood-thymus barrier here. Why would we want to mature immune cells away from exposure to the blood? From the general circulation. Because what happens if we had something in our general circulation, you had sepsis or something like that, we don't want to expose those immune cells to it. We want to mature them as the immune cells are supposed to be without influence of foreign antigens. So T cells are thymus, B cells are, where'd they come from? Bone marrow, perfect. So for our specific defenses, we're going to kind of coordinate these two things. We need our T cells for signaling and activating B cells. The important thing to know about B cells is these are the guys that build our antibodies. Your antibodies come from your B cells. All right, so you should know these types of T cells. We have cytotoxic T cells. These attack antigens physically and chemically. So they can bind to receptors. They can also secrete enzymes in, like lysozymes and things in to cause lysis of those cells. Helper T cells. These guys, you have to have your helper T cells to kind of coordinate your T cells and your B cells. So our immune system kind of has a built-in check and balance system. So if we realize, hey, we have something going here, and we want to activate T cells and B cells, these helper T cells basically say, hey, we need you. One checks the other. So it's kind of this double check system so that we don't get cells that kind of go haywire and attack cells that they're not supposed to attack. So this is kind of a check and double check system. If we don't have proper working helper T cells, we basically kind of shut our immune system down. Why? Because we don't have our check and double check and give the okay. If we can't check and double check and give the okay, the immune system does nothing. What's so significant about this is HIV virus, and this is important, you should know this. HIV virus attacks helper T cells. And so what HIV virus does is it gets in the cell. Most viruses do get inside a healthy body cell. So it takes longer to kick viruses, and antibodies can't attack viruses because they're hiding inside of normal body cells. So macrophages and things like that aren't going to detect them without some sort of a signal. But helper T cells are the ones that go, hey, we have an issue. They're sort of doing some surveillance, and they're telling B cells and T cells, hey, we need to work together to attack this. So HIV virus actually goes inside of the helper T cells, and it does reverse transcriptase to it. So basically it inserts its mRNA into helper T cells, and usually from mRNA we go to tRNA and we build a protein. But what happens with HIV virus is it takes mRNA and it goes backwards and changes the DNA from the HIV's mRNA. So it actually alters the DNA of the helper T cells, Now the helper T cells are like, oh, this is normal. This is what it looks like. And they don't detect that the HIV virus is in the body. Why? Because it has basically kind of hijacked all the helper T cells. So that's why with HIV, people die of really silly things, right? I mean, not that it's silly, I don't mean to discount it, but it's stuff that easily is kicked by our immune system or easily treated. Like, they'll get a common cold and they'll die of AIDS. Why? Because it shuts your whole immune system down. And if we take a common cold, but no one's doing anything about it, it can turn into this massive respiratory thing, and it kind of ends up killing you because we need these guys to launch an immune attack. If we take these guys out of the picture, they can't activate these guys that take over things. All right, regulatory T cells. These guys say, okay, that's enough. Pull back the troops. We've won the war. Everybody pull back, come back. So these kind of shut that immune response down. What happens if we never shut our immune response down? We have it kicked so into high gear that we would begin attacking good cells that don't need to be attacked. So these guys kind of call the end to the battle. Okay, we've won. It's time for everybody to stand down. That's what these guys do. And then these guys remember it for future battles. So if I catch something I've already caught, memory T cells will right away go, oh, that's what it is. They fire out the antibodies and they take care of it before we even notice anything. So we won't realize we have the flu virus. Why? Because we have antibodies that our memory T cells kick out, or they cause to be kicked out. All right. B cells make up 10 to 15% of our circulating lymphocytes. And the important thing about B cells is they secrete our antibodies. And this just, I'm not going to quiz you on how they subdivide. What do I want you to know? What each of these types of T cells do? What is their function? And then the B cells come from plasma cells. What do B cells do? They do our antibodies. All right, cell-mediated immunity. This provides cytotoxic T cells. Basically, think of these are going to be toxic to cells that need to be destroyed. So these guys find abnormal cells and pathogens inside of cells, and they're going to attack it using usually like lysosomes or some sort of an enzyme that does lysis. Antibody-mediated immunity, just know the definition. This is by the B cells. This is where we're going to kick out antibodies. Those antibodies are going to go take care of antigens that need to be destroyed and foreign pathogens. And do you remember in blood how we talked about with the antibodies? Antibodies basically like clog up receptors in cells. So those cells, and then they destroy them. So antibodies are much smaller than our immune cells are. And we kind of talked about how they can cross the placental barrier. So this shows adaptive. I mean, you can kind of walk through this. We can do cell-mediated, and we can do antibody mediated. So this is going to be cells that do phagocytosis. This activates T cells, which are going to secrete some sort of a toxin into it and destroy it. Antibody ones release these antibodies that will also clog up on these receptors and then destroy a cell. Antigens, what are they? They're a protein on the surface of a cell, right? We can have antigens that belong in the body. We can have foreign antigens, or we can display an antigen that says, hey, something's wrong with me. So all of those are still antigens. So not all antigens are bad antigens, but basically they become the targets for the immune response. That's how we know we need to do an immune response, is we look at the antigens on the cells. All right, I just want you to know these forms, these definitions. Innate is present at birth. Adaptive is what we acquire. Active develops after exposure to an antigen. Passive is when you transfer antibodies from another source. What's an example of passive immunity? You get bit by a black widow spider, that toxin's going through your body, and they go give you the antivenom for black spider bite or for a snake bite. They are injecting you with the antibody. Your body didn't have to work to build it. That's what passive immunity is. One of the things, not to be controversial, but one of the things that's driving me nuts about the COVID thing right now with the vaccine are people are like, well, you have to get vaccinated for active immunity. Natural immunity, if you caught COVID, that's still active immunity. It's active immunity as is vaccinated. So what is a vaccine? Usually with a vaccine, we take some form of whatever we're vaccinating against. Let's say rabies, we're going to give rabies shot to our dog. We take the rabies virus, we weaken it or kill it. In the case of rabies, they kill it, but there's still cells, even though they're dead, and they have those foreign antigens, and they inject that into the body. Well, the immune system's going around reading the antigens, and it's like, oh my gosh, we have this foreign antigen. It attacks it, it destroys it, even though it's already dead, but we build antibodies to it. Now we're not going to get rabies. When we look at like our COVID vaccines, that's what they do is they take some of the COVID virus, they weaken it, they put it into the body. Our body sees the antigens and goes, oh no, we have the COVID virus, and it builds antibodies. But if you caught COVID, your body did the exact same thing, right? It said, oh, I have this virus, and it goes in, it builds. So whether it's naturally acquired or acquired through a vaccine, it is all active immunity. Both of those forms are active immunity. So a lot of people try to differentiate natural immunity and they're calling vaccination active immunity. They are both active immunity.
You don't need both.
You should not need both. There's a lot of studies going out on COVID stuff. Yeah, I would say it's probably passive. I don't know exactly how that works, but I would guess it's passive. So. Well, I don't want to be controversial. Do we follow the science or do we do the news says? So if we follow the science, no, you shouldn't need it. So because acquired immunity, actively acquired immunity is actively acquired immunity. It doesn't matter where you got the antigen, your body activated antibodies against it.
But if you do acquire it, let's say if you had it before and you get it again, is it automatically gone from your body? Or do you have it in enough time that you could give it to someone else? With the vaccine or.
I don't know exactly what you mean. You
If I had or get vaccine, then someone gives it to me again. I would fight it instantly.
yes.
Oh, okay, that's
Yeah,
good.
it should. So I think some of the controversy around the COVID, I mean, there's a lot, but I'm like, it drives me nuts because I'm like, it's not science. I don't know. Anyway, but the whole thing is you do have some viruses or your antibodies will fall off a little bit. Honestly, they're still finding studies. In fact, the other instructor was just telling me, I just found a study where people who had COVID over a year ago, they're showing active antibodies from their active case of COVID. I don't really think they know yet on if we're going to need boosters. So if you end up, if your antibodies fall off, then yeah, at some point, maybe like the flu, where you may need a booster. And if it begins to mutate, now it really looks like a little bit of a different virus. So you may need something to it. But if you give a vaccination that has one form, and whether it's natural or if it's vaccinated, those are the antibodies you have until they fall off, which is what they do in flu. When you get a flu vaccine, they guess, we think this is the flu strand that's going to be out there this year. And they put those weakened antigens into your vaccine. Well, guess what? If it's not that right flu strand, what happens? You still might get the flu. So if you get little mutations or different strands, then it may not work 100%. But if your natural one matched to the vaccines, you should have the antibodies to it.
Is it like their cell is also recognized too? So one specific beat.
Correct. Yep. Yeah, it's very complex. We've made it sound really simplistic, but there's a lot of antigens. There's a lot going on there. Absolutely. I don't know. That's what I was wondering about. I just heard today. Anybody heard any much about the Delta? I just heard this is what I heard, and I don't even know because it's hearsay, but I heard it's more contagious, but less severe from their initial COVID. Has anybody heard anything otherwise?
They were all locked down.
And
just as many people died.
it was, okay, that's what I was curious about, because that's what I had heard. Anybody heard anything?
Yeah.
The vaccine's been effective to the Delta variant?
I heard that they're working on making the vaccine specifically for others variants out there.
To the D one. So, yeah, I don't know. And I wish that they had not made it such a political issue because I feel like it's really muddied the waters. If we could just go back to the science of it, I feel like we would get a lot better information. Maybe, or it could be that the levels fall off if the case isn't severe enough, but I think they work pretty well. So a good example of that is like chicken pox. Some people get, well, pretty much I think anymore, everybody gets the chicken pox vaccine. You can still sometimes get chicken pox, but it'll be like 2 or 3 pox versus a full outbreak, which is, you still have antibodies, but it wasn't enough of a response.
How about those people that are like, they get vaccine, let's say Hep B, to get their World Series Hep B and still have low titers?
Yeah, of the antibodies or, yeah, I don't know on that. I really am a big proponent of titer testing though. I think that's a really effective way to see where your antibody levels are at. I feel like sometimes, especially like in animals, if we take it to animals, we way over vaccinate. In my opinion, all of our animals, if you did titer tests, you would probably find, oh, they don't really need that yet. Not to say they don't ever need it again, but I feel like titer testing is a much better way to go than just constantly vaccinating. Well, and what they're finding is sometimes overdoing all of that causes its own issues. Some of the cancer issues, it's causing mutations. So I feel like that, especially in animals, has become an issue of the high cancer incidences in, say, dogs, is they're starting now to say we way over vaccinate dogs. So, I don't know, kind of interesting. The immune system's super complex. There's so much to it. And I won't pretend to be an expert on the immune system because I am not by any means. So it is so complex. All right, so I said know these, just know what they are basically, what is natural, what is artificial. What's the difference between artificial acquired active immunity and naturally acquired active immunity? Nothing other than the way that we receive the antigen. Is our immunity the same? Yes. It's just what form did we receive the antigen in? In natural acquired, we received it out there in the world in the environment somewhere. Artificial means we got some sort of a vaccination against it. Still, they should both be equally effective. Passive, again, this would be something that is given to us, antibodies that are given to us, like antivenoms, antitoxins, things like that. And we can also get those innate ones that are basically come from, we have innate non-specific, which are things that like skin barriers, but we also have our innate immunity that came from our mother. We didn't have to work for it. So active, we have to work for. Innate, we don't have to work for. So passive, again, we didn't have to work for it. This would fall under that. And then our active, we had to work through for it. Specific, this knows exactly what it's after. It recognizes that it goes after. This is going to be our T cells and our B cells. Memory are going to remember those. They're just circulating around to remember something when it shows up. Those are the ones that go get it right away by providing those antibodies when it sees something it knows. Tolerance, this is where the immune system ignores normal antigens. So we see things that are normal, that are part of our body. We're not going to go attack them. Antigen presenting. So we have cells that will engulf things, like a macrophage will take a cell in, it will basically chomp up that bacteria, that virus, and then it can present pieces of that antigen on its cell surface, which targets other cells. Hey, I have something wrong with me. Why? I just ate a foreign bacteria, and now that's going to launch an immune response. So that's what antigen presenting is. I'm going to skip these MHCs. I always say MHCs are basically your little antigens, your name tags on your cells. And I'm going to skip these because you guys are going to cover them and I'm not going to quiz you on them. It gets really complex in there. All right, so cytokines, these are just chemicals released by cells. We kind of talked about interleukins and interferons. Cells release them to basically, like interferons, we said it allows surrounding cells to know, hey, I've been attacked by a virus. So those cells can put out their antiviral proteins and kind of protect to themselves. But these are other ones, tumor necrosis factor, phagocyte activating chemicals, colony stimulating factors, and then just a whole class of miscellaneous ones. So these are all my chemical signals that help kind of alert or let other cells know what's going on with the immune system. Okay, that's all you need to know on those. What do cytokines do? They basically stimulate T cells. They're going to attract macrophages, which will help to engulf and destroy those cells. Those, I kind of mentioned this, they're going to present those antigens on their surface, which can activate like a cytotoxic T cells. Remember when I was telling you T cells and B cells work together in the immune system with our active immune system? And who facilitates a lot of this? The helper T cells. They kind of coordinate these T cells and B cells. I'm going to skip these guys. So helper T cells, I'll just talk about these guys. They sensitize B cells. B cells can't go active until helper T cells. So it's kind of a check system. The B cells are like, oh, I see this, I better go get it. But they have to stand there and wait until the helper T cells go, yes, you can go and get it now. So they may recognize something, but they can't kick off and go to work till helper T cells kind of say, yep, you can do it. It's sort of a check and balance system. So helper T cells are super important in activating my T cell, B cells. All right, plasma cells are going to secrete our antibodies. Memory B cells they have a memory of what they defeated, and then they can basically activate if they see it again, and we can take care of it really quickly. I know I'm going super fast, but I kept the test portion of this super simple. Now we talked about the titers. You guys all know what a titer is? It's just a test that reads your antibody level to something. Exactly. So if your titer levels are low, you would need another vaccination.
Chicken happened with my chicken pox. It went more off. They got one super young.
Oh, and so did you get full-blown chicken pox or just a light
No,
case?
she just saw that.
Oh,
They did the testing like pregnancy.
oh.
She's like, you could do
So they did a titer test?
it. They must have.
Yeah, they must have. I didn't know they did that. Interesting. All right. So before, oh, so what I want you to know, you don't have to know all these different antibodies, but just know that when you have a baby, they're going to inherit antibodies, different proteins from mom. This you should know. Stress and immune response. So what happens when we get stressed? You guys have probably all heard at some point or another, oh, I got sick. I was so stressed and tired and I had so much going on. It is true that your immune system kind of gets worn down from stress. So because stress will kick off that immune response, that it does that inflammatory response of the immune system. And so when we have high stress times in our life going on, part of your immune response is to secrete those glucocorticoids. You guys know where they come from. Where do they come from?
Yeah.
Yes, the fasciculata, because it's go find Rex, so it's the F, make good, the G. So it is the fasciculata, and they secrete those glucocorticoids. Cortisol is a good example of that. So if we weren't secreting glucocorticoids in a stress response, then we might see a worse response. We might not handle that stress. So when we secrete this, it helps just to handle those situations. It puts plenty of glucose out. Because remember, any kind of stress, your body doesn't differentiate stress from a fight or flight response. So when we have that fight or flight response, we're going to secrete those glucose molecules just to help handle it. Long-term secretion or chronic stress, you almost build like a tolerance to it. So you get that kind of an inhibitory response where your body doesn't really realize that anymore or recognize it anymore because it's there all the time. It almost like it becomes the new normal. Now that we do that, we kind of lower our resistance to diseases. What do glucocorticoids do? They depress inflammation. They reduce the activity of phagocytes in peripheral tissue, and they inhibit interleukin secretions. We said interleukin secretions basically are telling other cells, hey, we have a problem going on here. So that would kind of be part of that inflammatory response. So if we weren't secreting this, we'd see this high inflammatory response taking place. All right, what are some immune disorders? Allergies are, you can have a hypersensitivity or overactivation from all of the little pathogens that come in as allergies. So you might see a really high inflammatory response in your respiratory system, things like that, to the point where you might even have like an anaphylaxis response. Autoimmune disorders, these are going to be things like lupus, immunodeficiency diseases. So again, these are going to be like our allergens. And I would say just under these, you can get your, basically like your anaphylaxis, your overreactive response to these allergies. And that's what I was just talking about. It can be fatal. Obviously, one of the things is we do such an inflammatory response to the airway, the person actually can't breathe anymore. That's one of the bad parts that creates the fatal version of it. But we get lots of swelling, things like that. We can also go into anaphylactic shock. Why? Because of vasodilation. And this kind of just shows We get a lot of over-inflammation. Antihistamines. This is a time when, if you have an allergic response, you want to take an antihistamine, right? Right? Because it's, what is histamine? Inflammation, and we're going to de-inflame stuff. Some of our autoimmune disorders, rheumatoid arthritis, thyroiditis, type 1 diabetes. We talked about rheumatoid arthritis in 201, how we see loss or destruction of our joints due to autoimmune disease or disorders. And then we get our diseases, which is going to be things like lupus. AIDS is a viral autoimmune disease. Who does AIDS target? Helper T cells are the ones that get really attacked here. So what do we do? One, we can suppress the immune system. In some cases where we're like over hypersensitivity, in the case of AIDS, that you don't have an immune system. All right, what happens when we get old? We definitely get more vulnerable. Our immune system becomes a little less sensitive. It doesn't function as well. Our thymic hormone, so thymosin, reduces. Our thymus gland shrinks. Remember how we were saying you can't even see a thymus gland in adults? Like when we look at our gal over here, you won't be able to see her thymus. T cells aren't as responsive. We have less T cells. And then we see tumor surveillance cells don't recognize things as well. All right. That's super speed. On the immune system, the lymphatic system, I really went light on it. So it's not, you don't have to spend a ton of hours reading through all the details of that. Respiratory system, I do talk about in more detail. So let's jump into the respiratory system. I do think the respiratory system's a little easier, just in that one thing to know about the respiratory system, I kind of touched on a little bit of the anatomy today, but the other thing to know about the respiratory system is it follows concentration gradients. And so we're pretty familiar with concentration gradients hopefully at this point. And so this is a little bit easier. I'm sorry, what? Yeah, it follows the concentration gradient rules. Yeah, so what does that mean? Things flow from areas of high to low. So we're going to see oxygen, carbon dioxide, things are going to move down their concentration gradients. All right, so let's jump into this. All right, so I'll kind of go through the anatomy pretty quickly. Do you guys feel pretty good about at least the lungs and stuff that we talked about? So what is the function of our respiratory system? Obviously, it's going to be that gas exchange component. We're going to bring oxygen in, we're going to bring carbon dioxide in. The important thing to know, though, when we're breathing in air, is air pure oxygen? No, we're going to have lots of gases in the air. So what we have to start looking at is partial pressures of things. What does that mean? When I look at the air, I'm going to look at, hey, how much of that air is oxygen? How much of that air is water? How much of that air is nitrogen? Some of those things. So we are going to look at the concentration of that particular thing in the air versus the concentration in our lungs. If the concentration is higher in the air than it is in our lungs, it's going to move in. If the concentration is lower out here than in the lungs, it's going to move out. So we're going to be looking at concentration gradients. That's how things are moving in and out. Not only does it move into the lungs from the atmosphere that way, it moves from our bloodstream into our tissues that way. Where do we have the higher concentration of oxygen? Is it in the bloodstream or is it in the peripheral tissues? If it's in the bloodstream, we're going to move it into the peripheral tissues. And we're going to do the same thing with CO2. If I have a higher concentration of CO2 in my peripheral tissues as compared to my blood vessels, my CO2 is going to move into my blood vessels. So everything's moving down their concentration gradients, which makes it pretty easy, in my opinion, to figure it out. Just look at where's your area of higher concentration. All right, so a couple of things that, let me backtrack really quick. I kind of got ahead of myself here. Oops. This is all of the functions of your respiratory system. So we're providing a big surface area for gas exchange of our O2, CO2. We protect our respiratory system and we protect our lungs in our respiratory system. One thing you do need to know for both your lab exam and for your lecture exam is you need to know what the tissue types are found in these airways. So when I look up at my nose and I have epithelial lining my nose, what would my tissues be? What's my epithelial tissue type? Are they going to secrete mucus? Yes. Why would they secrete mucus? Yeah, so I can trap little particles, right? Do I want all the little particles floating around the air, the dust, the dirt that I'm walking through to go down to my lungs? No. So as you're studying this, make sure you study the tissue types found in each area. If it has direct contact with the outside world, it's going to have mucus, and we're probably going to find some amount of cilia. The mucus is going to warm and humidify the air, and it's going to trap particles. So those are important. Warming and humidifying the air also protects the lungs. So we're looking for warm, humidify, and protect. How are we protecting it? This is part of the protection, but also that trapping and moving particles. So as you study for this, for both lecture and lab, you want to start looking at those different types of tissues found in every area. And we'll kind of talk about them as we move through here. So we're protecting those respiratory surfaces from dehydration, temperature changes, real cold air, more than even real hot air, but hot to some extent. We're also producing sounds, right? Our respiratory system, we talked about like the nasal conchae in 201. They're covered with epithelium. What are they doing? They're resonating sound. They're helping with our phonication, which is your sound. They also detect odors in our nose, which are actually tied to our taste buds, right? You guys remember that? Okay, upper respiratory system. Super important that you know the difference between upper respiratory system, lower respiratory system. I'm probably going to ask you about that. So what is your upper respiratory system? It's basically everything above your voice box. Our voice box is where the last of the pharynx is. It becomes, your voice box becomes your larynx. At that point, we're into new areas. So pharynx, remember that term pharynx is shared tubing. Who's sharing this tubing? What systems? Digestive and respiratory. If both of these systems are sharing it, we call it the pharynx. As soon as they split into their own tubing, we're going to change the name. So respiratory system is going to own the larynx, the trachea, the bronchi. That goes with my respiratory system. But up until that split, it's all pharynx. The digestive system then becomes the esophagus, which is the start to the true digestive system. So upper respiratory system is everything from the nose, which is our respiratory. We can breathe through our mouth, but we really don't consider our mouth part of our respiratory system. We consider it digestive system. So everything from the nasal area through the shared tubing with the digestive system is our upper respiratory system. What's our lower respiratory system? Where we branch into just the respiratory system. So it's going to be the larynx, your voice box, your trachea, your bronchi, where they split, bronchioles, alveoli. Yes. Your epiglottis is the division trap door. I would probably put it with the larynx and put it with the respiratory system, because it really kind of sits right over the trachea and covers the trachea. So good question. All right, so this shows, here's my whole nasal area. This is all pharynx. This would be nasopharynx, oropharynx. Then we get down, here's my epiglottis, covers the trachea. when we swallow, and things would go back into the esophagus. If we're doing respiratory things, the epiglottis stays open and stuff will route down into my lower respiratory system. All right, respiratory tract is the conducting portion. What does that mean? The part that's moving the air in and out. So this is the conducting portion is from the nasal cavity to the large bronchioles. The respiratory portion would be the bronchioles and alveoli. So this is the very end. When you see the little grape clusters, you guys remember seeing all the little illustrations of the little grape clusters? This would be my bronchioles here, and this would be my alveoli here. So now, this is where my exchange is going to begin to take place. So this is my respiratory. Conduction is just moving the air. Respiration, we get down into the area where we're going to be exchanging. Specifically within this respiratory portion, the alveoli is my exchange. These alveoli, these little grape cluster sacs, are loaded up with capillary beds come into each of these alveoli. And this is where we begin to look at the concentration gradients of my partial pressures. So how much oxygen do I have? How much carbon dioxide do I have? And we begin to exchange based on those concentration gradients. Excuse me. All right, respiratory mucosa. What does mucosa mean? It's the layer that secretes mucus. Anytime we use mucosa, we're talking about a layer that is secreting mucus. It will be an epithelial layer. So what is my epithelial mucus secreting portion of my respiratory tract? It's basically going to be all of this in here that is doing what we talked about: warming air, humidifying air, protecting from particles, things like that. So your epithelial tissues are your respiratory mucosa. They're secreting mucus to protect the airways. All right, when we get in the upper portion of our epithelium, we'll talk about the specifics as compared to down in the exchange portion. All of our epithelium has to sit on a connective tissue, correct? And it is our lamina propria. Specifically, it's our loose areolar connective tissue. We have a respiratory defense system. Part of this is moving those particles. So if you guys remember in the trachea, we found the ciliated pseudostratified columnar. And the mucus traps the particles, the cilia sweeps it up and out. This is part of my respiratory defense mechanism. I'm going to trap particles in my mucus. I'm going to sweep the mucus with the trap particles up and out. What is my hope? I don't get pathogens. I don't get particles down in my lung. Do our epithelial layers and our connective, our lamina propria layers have immune cells in them? Yeah, especially areolar. Remember, loose areolar had macrophages, fixed macrophages, free-floating macrophages, mast cells, all of those guys. Microphages, these are all found in this loose areolar. So they are going to be part of that defense system as well.
She did the rest.
The trachea, or
Pseudostratified?
yeah, ciliated pseudostratified or pseudostratified ciliated columnar. Uh-huh. All right, so upper respiratory system has mucus glands that discharge right onto the epithelial surfaces. The conduction portion, the lower respiratory system, has a lot of smooth muscles. What can they do? They can dilate, they can make those bronchioles smaller, they can make those bronchioles bigger. Was everybody okay? All right, and this just shows my epithelial surface here. This is my ciliated pseudostratified columnar sitting on my loose areolar connective tissue, which has all those mast cells, macrophages, all of those guys. And this shows a picture of them. So these are my ciliated pseudostratified. There's my cilia. Here's my mucus. And as stuff comes, it can trap in the mucus. The cilia secretes it up. We have mucus secreting cells here. We also have these deeper mucus secreting cells. This is my loose areolar connective tissue, which would have a lot of defense cells in it. And this is a zoom in of the cilia and the mucus. So this walks you through your tissues. We just said the upper is the pseudostratified ciliated, you can say ciliated pseudostratified. Inferior portions of the pharynx, right above our esophagus, these guys become stratified squamous. What did stratified squamous do? They do protection from abrasion, right? Why would the lower portion of my pharynx be stratified squamous? Yeah, when I'm swallowing, this is shared tubing with my digestive system, so I have to protect it against abrasion. So the bottom part of the pharynx before a split is going to be more of a digestive system protective tissue. Then we get into the superior portion of the lower respiratory. So we've made our split and we're into the trachea, and we're going to be back to our ciliated pseudostratified columnar that can trap and sweep those particles. When we get down into the smaller bronchioles, we're going to have cuboidal cells. So we're down in this area here, cuboidal cells with cilia, scattered cilia. Hopefully we stop to the big particles up here. If we haven't, we can still move down, hopefully trap them here. Is everyone good?
Well, no.
Yeah, so we start with ciliated pseudostratified columnar. When we begin to share with the digestive system, we switch to more of a digestive tissue, which is the stratified squamous. And then when we go back to true respiratory, we go back to the ciliated pseudostratified. Why? So we can trap and sweep, and we have that mucus. Then we get down in here, we're cuboidal with those cilia. Cuboidal, remember, creates a lumen, and we put that smooth muscle around there. We're going to have the ability to change the size of the lumen using the cuboidal. All right, alveolar epithelium. This is down here. These guys, we're going to be doing quick gas exchange. So what kind of tissue are we going to see? Simple or stratified? Simple squamous, right? So single layer flat cells allows rapid movement. If I have to go through 10 layers of cells, that's not going to be very fast, right? If I go through one layer of cells, it's going to be really fast to movement. So when we get down into the alveoli down here, we're going to see simple squamous. Why? Because gas can move through really, really quickly. Do we want gas to move fast? Yeah, absolutely. That's the whole point of this, right? All right, respiratory defense. Part of it's filtration. We're going to filter it through hairs in the nose with mucus. We get down here, we have the cilia that's trapping and sweeping things. And then if we get particles or pathogens that make it all the way down into here, they make it past all those ciliated mucus areas, we actually have macrophages down in here that can destroy pathogens, destroy dirt. And those are called my alveolar macrophages. Ideally, this stuff never makes it to here, but in case it does, we can take care of it. What you'll see with some people who say coal miners that are breathing in that chronic dust all the time, maybe people who work in construction, drywallers who are tearing off drywall, something like that. The dust is so overwhelming, we can't stop it in the upper respiratory system. Sometimes you can kind of overwhelm this lower respiratory system, which can cause all kinds of breathing areas. We begin to affect the surface area for gas exchange. All right, nose, just know it's the nares. What kind of tissues are we going to see up there? We're going to see mucosal tissues still. They're going to secrete mucus. And we have the hairs that are going to trap things on the mucus. Nasal septum, you guys know what this, I'm not going to get into all the nitty gritty details of all this. You guys know most of it. What is the whole purpose of these nose openings? It's to trap particles in the mucus, warm and humidify air. What is warming and humidifying air doing? It's protecting our lungs down here. We want it warm. We want it humid before it gets down into there. It also helps, remember, with that absorption of those odorants into the epithelium for the sense of smell. Yes.
Your,
I have heard that before. Yes, you can damage your capillaries with too cold, too hot. Too hot, I'm talking like you're in a fire and you breathe in something that hot. If you're out walking on the desert and it's 120 degrees, your capillaries are probably fine, but the rest of you's not fine, but your capillaries are fine until the rest of you begins to fail. But yeah, cold air is really hard on this. It does hurt your lungs. The other thing you're going to see, and I don't get into a ton of detail on it, but when we talk about saturation curves, basically, what does that mean? How much oxygen are we putting into tissues? Are we saturating our tissues with? The warmer the air is, the better our saturation levels of oxygen are. So when you get in really cold weather, cold air, we don't pick up oxygen and saturate our tissues at the same level. So sometimes with the really cold, you'll see hypoxia in people because we just don't absorb and move oxygen at the same rate, and it's really cold. Pallets, we know what those are. I'm gonna skip through all this stuff. This is at Body World. Have you guys ever been to Body World? Oh, it's so cool. They have this exact thing at the Body World exhibit. It's the German guy who takes the bodies and he does all the poses with them. It came to Denver to the Museum of Natural History. Yes, Las Vegas has one. The one in Denver was so much better than Las Vegas is, but it's really cool. It's called Body World. They have one where they do a cross section of a really heavy person and they show the layers of fat as you go through all the levels. They have one that they do all the blood vessels of the face and it's just the blood vessels and you can see the whole face and all the detail. It's really cool. If you ever get a chance to go to a Body World exhibit, in my opinion, it's real worth it. My kids thought it was super creepy, but I could have stayed there for hours. It was so cool. All right, so here is our nasal area. This becomes our pharynx back here. Again, here's our epiglottis. This is our trachea. This is our esophagus. Nasal mucosa, I've talked about. Nosebleed is extensive vascularization of the nasal cavity. So you get too much blood flow going. Sometimes you get a little damage to that nasal area. Oftentimes with dryness, it gets too dry and you'll get nosebleed. Pharynx, nasopharynx, oropharynx, laryngopharynx. Nasos up the shared tubing behind the nose area. Oros, the shared tubing behind the mouth. Larynx, the shared tubing right at the larynx level, right above the larynx area. All right. You do not have to know the cartilages of the larynx, so I'm going to skip through that, or the hyoid cartilages. I'm going to skip through those, or the ligaments. This shows your vocal cords. It's kind of interesting. If you ever want to see a cool video, in my opinion, go to YouTube and they have one of, oh my gosh, I just forgot his name. He did Steven Tyler. That's who it is. They have where he does singing and he hits those really high notes. And they've put a little camera down and they show his vocal cords. It's really cool to see what your vocal cords do. So they have him doing like the whole gamut of sounds and singing some of his super high songs and stuff. So it's kind of interesting to watch what the vocal cords do. And this is exactly what they look like. I think that's what my video link was to Steven Tyler singing. So, but you guys can find it on YouTube if you want. Let me skip that. Trachea, it's your windpipe. we are going to split our trachea into our right and left bronchi. The split, right where they split, is called the carina. So that is our split area. I don't know why it doesn't have it on here. I think we might have it on our lab list. Carina. This is right where your bronchi split from your trachea. We do secrete mucus in this tracheal area. I'm going to skip these, but this shows, here comes our larynx. We're going to go down our trachea. This split is called the carina. There it is right there. And we're going to split into our right and left bronchi. This shows your esophagus behind your trachea. And this would be a person like bending down or looking at that cross section. This would be the anterior. This would be the posterior. All right. Basically, I would like for you guys to know the branching, how they branch. Basically, it goes from the bronchi to the lobar bronchi to the segmental bronchi. You don't have to know the numbers in the right lung versus the left lung. But if we were to draw basically a flow chart of the branch, it's going to start at that main bronchi. We're going to go lobar bronchi, segmental bronchi. We talked about the carina. This shows a nice picture of it. So here's our trachea. Here we split into our main bronchi. Then we can go into our lobar, which are basically our lobes. They go into the different lobes of the lung. And then into our segmental, which are the different segments in the different lobes. And this shows it kind of branched out. Trachea, bronchi, lobar, segmental. Then we get into our small bronchi, bronchioles, into our alveolar. So this is your exact flow chart right here. Bronchi, lobar, segmental. They just get smaller into your bronchioles, into your alveoli. Is everybody good? All right. And this just adds everything. I really like this picture because you can see how extensive the capillary bed is at all the alveoli. Obviously, we're going to have lymphatic vessels, right? Because we said lymphatic vessel mirror your other vessels exactly. So we're going to look at these alveoli. What is bronchitis? Just know, well, and I kind of skipped this. The only thing really to know that when we start up here, we have a lot of cartilage. By the time we get down low, we're more into the muscle area and we can contract, dilate, and contract those using those smooth muscles. What is bronchitis? It's just inflammation and constriction of the bronchi due to infection. So when we start splitting and getting down into our lungs, if you get an infection, it can cause fluid buildup, inflammation. Usually bronchitis is accompanied with really bad coughs. Anybody need more time? All right, terminal bronchioles, these are the end where we now dump into our alveoli. These are dominated by smooth muscles. There's no cartilage when we get down into the bronchioles. Who controls the diameter of that? They're smooth muscles, so they're controlled by the autonomic nervous system, right? It happens automatically. We don't have any control over it. Bronchodilation, this is, we're gonna enlarge those, right? We're gonna dilate it, make them bigger. Bronchoconstriction, we're going to squeeze those muscles, we're going to make our lumen smaller. Why would we do that? Histamines, these are parasympathetic usually. If we're relaxed, we don't need as much airflow to our lungs, we might bring those down. It's a parasympathetic response, but we'll also see this with a histamine and over allergic response like anaphylaxia. It'll constrict those bronchioles to the point where a person's actually can't get a lot airflow. What is asthma? Extensive stimulation of our smooth muscles, which is going to restrict airflow. All right, I'm going to jump down into the alveolar sacs. Just want you to look at a picture of the capillaries, and then we're going to jump into how this works. So this shows, we saw a picture almost identical to this in 201. All right, we have two types of cells that are working in the alveoli, aside from the macrophages, which we know what the macrophages do. What are they doing? Destroying pathogens and particles that make it to the lungs, right? So that's, these guys are immune, but we have pneumocytes type 1, pneumocytes type 2. These guys do actual gas exchange. It is simple squamous, we know that, right? So we have that rapid gas exchange. The pneumocytes type 2 produce surfactant. And I kind of talked about this last week when we talked about residual volume in the lungs. What did I say residual volume was? Yeah, after, even if I forcibly exhale, I can't exhale that air out of my lungs. Why? We said it's the amount of air we need to keep the lungs inflated, right? Because what did I say? If we collapse the lungs, what did we have to do? We had to overcome the water, the surface, the polarity of water molecules in the lung, which would have to pull that water apart. What the pneumocytes type 2 do is they produce a surfactant, which is basically a chemical that helps to overcome that polarity of my water molecules anyway. But residual volume keeps our lungs inflated. Pneumocytes type 2 produce surfactant, which helps us to overcome that surface tension. All right, and this shows your alveoli. You can see our pneumocytes type 2 in here. Here's our macrophages. Surfactant is an oily secretion. And again, it's going to reduce that surface tension of the moisture in my lungs. The water molecules in my lungs are not going to stick together because of this surfactant. So what are some respiratory distress things we see? If we collapsed an alveoli, now we have to overcome that surface tension to reinflate that alveoli. So respiratory distress, if we collapse a lung, now we've collapsed, we've taken that residual volume out. But it can be caused from injury. It can also be caused by not producing enough surfactant. So when we get down close, the water molecules will actually stick together and take the residual volume out of my alveoli. So we need two things to keep our alveoli full and inflated. We need the residual volume of air, but we need the surfactant so that the whole of the polarity of my water molecules don't collapse my alveoli. All right. We have an endothelial layer in our capillaries. Remember that simple squamous? Why do we want simple squamous? We can move things quickly, right? All right, so this is showing my red blood cells. This is showing my alveolar space. So basically my capillary has a single layer of flat cells on its surface, and now I can easily change things back and forth from my bloods, from this would be my alveoli into my blood, or I could trade, if I had CO2, I could trade CO2 from my blood into my alveolar space. All right, so the distance is really short. We don't have very far to go is one thing. O2 and CO2 are small and lipid soluble. What does that mean? It can go right through the cell membranes. I don't have to be transported across, right? We said anything that is lipid soluble, can just diffuse right through the cell membrane. O2 and CO2 go right through the cell membrane. So we're going to see fast movement based on concentration gradients. All right, pneumonia is inflammation of the lung tissue, causes leaks in the alveoli. So you actually begin to fill those alveoli areas up with fluid. If I'm full of fluid, I can't move that gas across there very easily. So it compromises that gas exchange area.
It's not just water.
It is not, it's a fluid type substance. It does have probably some amount of water in it because it's a plasma-y based type thing.
Like people explain it like it's water in the lungs.
Yeah, it is what it's like because it's just basically you're drowning
Yeah.
because we've taken the air sac space away and put fluid in it. So we're not going to see the gas exchange. Okay, I talked about this in lab, so I'm going to go kind of quickly here. We have right lung has 3 lobes, superior, middle, inferior. We have a horizontal and an oblique fissure. Horizontal is the one that goes across, oblique goes at a slant. Right lung is wider and it's displaced upwards because of the liver. Left lung was longer, but it only has the 2 lobes. We saw the cardiac notch. What was the function of the cardiac notch? Space for the heart. You got it. Hilum or hilum, what was that? Yeah, it's like the pelvis where stuff goes in. My pulmonary artery, pulmonary vein, and my bronchi go into my hilum, along with the lymphatics, because lymphatics always follow vessels, right? All right, so this shows my 2 lungs. I'm not quite sure why his are so black. Maybe he was a smoker. Yeah, because those were pretty black. Here's my superior, middle, inferior. Here's my horizontal fissure, oblique fissure. And this side just has that oblique fissure. Here would be our cardiac notch. Here's our hilum, basically this pelvis. All right. What is a pulmonary? So blood pressure in the pulmonary circuit is lower than the systemic. We know that, right? The heart pushes hard and pushes blood out into the systemic. It has to make it all the way to your toes. Does your heart have to push very hard to go to the lungs, which are right beside it? No, it doesn't. So we're going to see a lower systemic pressure in our pulmonary cycle than we see in our systemic. I think I just said lower systemic, but I meant lower pressure on the pulmonary side versus the systemic side. What is a pulmonary embolism? This is basically when you get a blockage or you lose that flow of blood through the pulmonary artery. This is what an embolism is. Typically, it is blocking of that. A lot of times people will get like blood clots. They'll talk about like throwing a clot to their lungs, things like that. This is what they're probably talking about if they're using that term. So what happens if I stop blood flow to my lungs? Nothing good. Can that blood go pick up oxygen and come back to the heart? No, I shut off my blood supply right there. So the rest of my tissues are going to become hypoxic, including my heart, right? Because the heart is getting blood when it's come back from the lungs through my cardiac cycle. All right, we have 2 pleural cavities. The mediastinum or mediastinum was that cavity right up the center. You have your right and left pulmonary cavities. Oops. All right, external respiration. I'm going to ask you the difference between external and internal respiration. External is the exchange of O2 and CO2 with the external environment. So when I'm breathing in, I'm breathing out, that's external respiration. And I'm taking it in, I'm exchanging it at my lungs, that is external respiration. What is internal respiration? It's when I take it from my blood and I put it in my tissues. So internal respiration doesn't necessarily, well, it doesn't have direct contact with that conducting portion or respiratory portion of my airway. Internal respiration is all about from blood to cells, cells to blood. And we're going to follow our concentration gradients when we look at the exchange in the lungs. We're also going to follow our concentration gradients when we look at exchange with our blood and our cells. Is everybody good? All right. So external respiration. First, I'm going to breathe. We breathe using concentration gradients, and I'll talk about that. Pressure. We use pressure gradients. We are going to move that gas in our alveoli between our lungs into our bloodstream. And then we're going to transport, once it hits our bloodstream, obviously we're going to transport it to those cells. Everybody good? Okay, so I really like this picture. This shows external respiration here. I breathe it in, I trade here at the lungs. Where am I trading? I'm taking waste, CO2 from my blood, I'm sending it to my lungs. I'm taking oxygen from my lungs, I'm sending it to my blood. That is external respiration. Internal respiration is this blood has now left the lungs, it's traveling around the body. And as I go, I diffuse O2 out of my blood into my cells and my tissue. Waste from my cells and my tissues, like CO2, goes into my blood, which eventually will make it back to its lungs to do external respiration. So this exchange here is external respiration. This exchange here is internal respiration. Hypoxia just means low tissue oxygen levels. Anoxia means I have no oxygen to my tissues. We have cut the oxygen off. This just means low oxygen. Are you guys okay? Do you need a little, or is everybody okay? Everybody good?
You're good.
Okay. I'm fine too. All right, now this term, pulmonary ventilation, this is breathing. This is the physical movement of breathing. And we're going to look at atmospheric pressure. What is atmospheric pressure? It is the weight of the Earth's atmosphere on us. Atmospheric pressure here is not the same as it is in Charleston, South Carolina, right? Or Newport Beach, California. Those guys are at sea level. They have a higher atmospheric pressure than we do up here at, I don't know what Colorado Springs is. What is it? 5,000 feet? No, probably higher than that. No, probably about right. Anybody know what the, I know out where I live, it's 7,000 feet, but yeah, the Black Forest area is around that, but I think Colorado is down 5,000 or 6,000 feet, but that's still high compared to sea level, right? Big change in atmospheric pressure. So we have to look at atmospheric pressure. The more weight we have above us, the easier we push things in. The less weight we have above us, the harder it is to move things in, because we're moving with the pressure gradient. If we don't have as much weight pressure, we're not going to bring it in as easily. All right. Here's, just to boil this down, basically we're looking at as we put molecules together, it creates more pressure. So if I took a container and in my container, it's closed, it has a lid on it. We'll put a little lid on it here. And if I had, I don't know, 6 molecules in this container, they have some amount of pressure on them, right? Because they're bumping into each other and they're moving as they bump into each other. So they're doing this random movement. If I take this container and I make it now much smaller, but I have these same 6 particles. Is there more pressure on it? Are they going to bump into each other more often? They're not? Yeah, they're going to bump in, which actually creates more pressure. It's kind of like if we took our hallway down here and we put in our hallway 10 people out in this hallway and they're passing each other in the hallway. That's all good and great, right? We see you coming, we step around you, we go the other direction. But now if I took my hallway and I made it a third of the width with the same 10 people, we have more pressure like to move away from each other, right? So it's the same kind of idea with our container. If we have a container with 10 molecules and my container is this size, if I do this with my container, I put more pressure on it, right? Because my molecules are going to bump into each other. They're going to want to move away from each other. If I make my container this big, I put less pressure on it, right? So our external respiration is all about the size of our container and how I put pressure on my molecules. So I'm just going to skip the Boyle's law and show you this. When we do external respiration, what we really are doing is we're changing the size of our container, which changes the pressure on our molecules. So if I have a certain amount of pressure out here in the atmosphere and have a certain number of particles in my lungs, which is a pressure, and this is the size of my container, and now all of a sudden I make my container this big, and I had this much pressure up here, but now, so I was at equilibrium. I had a certain number of particles here, a certain number of particles here, but now all of a sudden I make my container much bigger. Now where do I have more pressure? My container got bigger. I have more pressure here, right? So stuff's going to want to flow into the container. If I have a small container and I have a lot of particles here that are being squished more together than out here, where are my particles going to want to move? They're going to want to move out because there's less pressure out here than there is here. When we actually breathe, what we're doing is we're basically putting particles in our lungs. We're comparing the number of particles to the atmosphere, and then we're changing the size of our container. If we make our container bigger, we've taken the pressure off, stuff's going to want to come into it. If we make our container smaller, now we make all these particles bump into each other. They would have more room out here. They're going to want to move out. How do we change the size of our container? We do it by changing the size of our chest, of our breathing space. So when we inhale, we expand our rib cage, we expand our lungs. How do we expand our rib cage? Our diaphragm. So here's our diaphragm. It basically is the biggest thing responsible for changing the size of our container. Here's our little guy. Here's our diaphragm. When we breathe in, we contract our diaphragm. When we contract it, it actually goes down like this. Did I just make my container bigger? Yeah, and maybe I had, I don't know, 4 or 5 particles here. Let's have 4 or 5 particles out here. But now they have more space here. What are they going to do? There's less pressure here than here. They're going to flow into the container. Other things that help to enlarge my container are my external intercostals. Do you guys remember that from 201? They originate, if this is my ribs, here's my second rib. They originate on the bottom of this, the top rib, and they insert on the bottom of the below rib. When they contract, they lift and expand the rib below them. Changes the size of my container, right? It makes my container big. So if I have a certain number of particles and all of a sudden I make my container bigger, there's less pressure, stuff's going to flow in. Now can I change the, once I brought all these particles in, I'm going to make my container smaller. How do I make my container smaller? I'm going to relax my diaphragm, which puts more pressure on these particles. Now this container is smaller than out here. Particles are going to move out. What makes my container smaller? I relax my diaphragm, and your internal intercostals, those guys go this way. They go from here up to here. They pull the top rib down. So they pull the rib above, down and in. They basically take my container like this and turn it into a container like this. Now there's a lot of pressure on those molecules. They're going to want to flow out. So external respiration is all about, we change the size of our container. Wherever the pressure is less on the molecules, that's where the molecules are going to go. All right. And this kind of shows, here's our diaphragm. This shows some of the muscles. There's a lot if you get into forced inhalation, forced exhalations, your sternocleidomastoid, why it's grabbing your clavicle and your sternum, which is helping to expand my container. My scalenes, your scalenes go down the side of your neck. They attach to your first rib. So again, they're lifting that rib cage. Pectoralis minor goes from your ribs to your scapula. It helps to elevate ribs. Serratus anterior, all of these guys. But who are my primary ones? The ones we just talked about. External intercostals and your diaphragm. They are the ones that do the bulk of changing the size of our container. All right, exhalation. Who's our primary one? We're going to relax our diaphragm, internal intercostals, and rectus abdominis, even as we squeeze the ab in, pushes the diaphragm up, makes my container smaller. All right. Who's the major muscle that does inhalation? The diaphragm. And the external intercostals. Exhalation is going to be internal intercostals, compress the ab, move the diaphragm. Forced breathing is called hyperpnea. I don't know. I can never say that word. Paninos. Aren't we all hungry? Let's go to Paninos. Quiet. We have quiet breathing, forced breathing. What is this? This involves active inhalation and exhalation. What does that mean? I'm engaging all those various muscles when I inhale and I exhale. I'm pulling all those accessory muscles in. It's not just those main ones. Is everybody good? Quiet breathing is going to be our costals and our diaphragm. Elastic rebound, these are the other tissues that participate, things like fascia, things like that, that are going to have some elastic fibers that just recoil. All right, pressure changes. This is super important. Please know this number right here. Normal atmospheric pressure is 760 millimeters of mercury. You should know that. That is how much pressure is out here in the air. Ours is not this high. Why? Because we're at high altitude. But this is the normal working number. All right. Intrapulmonary pressure is also called interalveolar pressure. And it's basically, we're looking at our atmospheric pressure. Again, kind of think about in terms of size of our container. I'm not going to quiz you on these, just kind of know what it is. Intrapleural pressure is the space between my parietal and my visceral pleura. So I have my lungs and they're covered with a visceral tissue. And then remember, my lung cavity has a parietal tissue. There is a pressure between my lungs and the cavity. That's what we're talking about, is intrapleural pressure right here. This always stays below atmospheric pressure. When we get some sort of an injury that goes all the way into the thoracic cavity, into that pulmonary cavity, it can mess with this intrapleural pressure, which can cause collapse of the lungs, difficulty breathing. Why? Because how do I breathe? I'm going down pressure gradients. If all of a sudden I change the pressure, it's going to affect my ability to breathe. All right, what is a pneumothorax? This is what I was just kind of talking about. It's when air enters the pleural cavity. And this can be due to injury or rupture of an alveoli. So it doesn't have to be injury. It can come from an internal issue with the alveoli. And this can collapse my lung. Why? Because I get this really high intrapleural pressure, which is now squeezing down on my alveoli, can collapse those alveoli due to my pressure gradients. So what do they do? You always see it like on Grey's Anatomy, they get that, they take a little tube, yep. Why? They let that pressure off and then they seal it with airtight. Why? Because now we can't add more pressure in there. All right, what is resistance? This is basically how hard is it for the air to flow into the alveoli. How can we change that? Change the size of the bronchioles. Compliance is how expandable our lungs are. If I get damage to connective tissues around my lungs and they get scar tissue, they may not stretch and rebound at the same rate. I would call that low compliance. What are things that affect compliance? It can be age, it can be diseases, injuries, exposure to chemicals, exposure to pollutants, things like that. Surfactant production affects compliance. So if I don't have enough surfactant, I'm not going to be able to expand and come back. And then mobility of the thoracic cage. If I have a broken rib, am I going to want to move my rib cage to change the size of my container? No. And that's one of the big complications of broken ribs. They usually say, I think it's if you break 3 or more ribs, they actually hospitalize you. Why? Because people don't want to move their chest cavity because it hurts and you don't get enough gas exchange and pneumonia from that lack of movement, and clearing those lungs out is actually a big complication of multiple broken ribs. Why? Because we don't want to move that rib cage. It goes back to this compliance. All right, these I'm going to skip through because we covered them in lab when we talked about our lab last time. So I'm just going to kind of flip through these. If I didn't already talk about it, you don't have to know it. All right, gas exchange. This is all based on partial pressures. So let's look at partial pressures. We're going to move down our concentration gradients. We're going to split those concentration gradients. When I say atmospheric pressure, what did we say it was? Yep, this is it. Is that all oxygen? No, some portion of that's going to be oxygen. We're going to have nitrogen. We're going to have various things in that. Here's what we have in all of our atmospheric, that 760. Nitrogen is actually the largest amount of what is in your air is nitrogen. It's 597. So if I take this 760, 597 of it is nitrogen. Are we inhaling and exhaling nitrogen? No, we don't even care about it at this point, right? Oxygen is 159. Water, we have water in our air, 3.7. Carbon dioxide, 0.3. So nitrogen is overwhelmingly the most abundant gas in our air, but the second thing is our oxygen. These are our partial pressures. These are the parts that make up my total pressure. That's what your partial pressure is. And this is what we're going to be looking at for our concentration gradients. Is everybody good? All right. Henry's law, you don't have to know the law, but I will just touch on this for a minute. Temperatures can affect how easily things move back and forth. And I kind of talked a little bit about that. Under warm temperatures, we move oxygen at a faster rate, which makes sense, right? Because who's our number one heat producers? What raises our internal temperature? Skeletal muscles, which means we're probably moving our skeletal muscles. We're creating heat. If we're moving our skeletal muscles, we're using up oxygen. We're going to want to saturate with more oxygen. So warmer temperatures typically encourages oxygen to move. With this talks about, this is just kind of showing atmospheric pressure in a liquid. So if we have a certain amount of atmospheric pressure on particles, we're going to diffuse it into our liquid based on the amount of pressure. If we take this off, it's going to diffuse out at the same amount of pressure. So we'll see those particles push in or come out. When is this also become an issue when we look at like scuba diving? When you go scuba diving, we put not only the gas pressure, now we put all the pressure of the water on top of you, and we diffuse things in at a pretty high rate. One of the effects of the bends is when we come up, if we don't stop and sort of decompress, those gases will bubble as they come out of the blood, which causes the bends. So this is a really big deal when we talk about things like scuba diving and stuff like that. CO2 is highly soluble. O2 is not as soluble in water. Nitrogen has very limited solubility. And this shows a pulmonary vein. This would be my partial pressures. I'm not going to quiz you on these. All right. So gas exchange, basically what I want you to know about gas exchange is CO2, O2 is lipid soluble. What does that mean? It moves through cell membranes like instantaneously. It's a simple squamous epithelial cell. Again, it's going to move through instantaneously, and it is going to follow its partial pressure. It's going to go down its pressure gradient. We have a high surface area, so we have lots of area for these molecules to exchange. All right, that's our external respiration. What is external respiration again? Yeah, breathing in, exchange at my lungs, right? Into my bloodstream. What's internal? At bloodstream to tissues, right? Or cells. Okay, so blood arrives in the pulmonary arteries, which is it oxygenated or deoxygenated in my pulmonary arteries? Deoxygenated. So I can also look here. I have a low partial oxygen pressure. It's my deoxygenated blood. And I have a high CO2 partial pressure. That makes sense, right? It just came back from the body where we were dumping all the CO2, taking all the O2. All right, with our concentration gradient, what we're going to do when we get to the lungs, we've just breathed this oxygen in. We said atmosphere, we've hardly had any carbon dioxide in the atmosphere, right? It was 0.3. So when we breathe in this air in the lungs, it's going to be low in CO2. So where's my CO2 going to want to go? From my blood into my lung space, right? Because that's where I have less CO2. And as I go high O2 in my lungs as compared to the O2 in my blood, O2 is going to want to go into my blood. So they just follow their concentration gradients and they do this quick switch right here. So O2 goes into the blood, CO2 leaves the blood and goes into the lungs. And for a brief moment, it reaches equilibrium on its partial pressures of O2 and CO2. Does that make sense? All right, so we're just following our concentration gradients. Is everybody good? All right, when we get into internal respiration, so now this oxygenated blood has traveled in the bloodstream to the tissues. The tissues are constantly using up oxygen, right? And when they're using up oxygen, they're doing cell respiration. What's a byproduct of cell respiration? CO2. So these tissues have low oxygen while we're using it up. What are we kicking out when we use it up? CO2. So they're low O2, high CO2. My blood just got there and I got the oxygen from my lungs. So now it's high O2, low CO2. We switch with our tissues because of the concentration gradients. So we just do this switch. You don't have to have the numbers. I just want you to understand the concentration gradients. Anybody not okay with those? there's a great picture. I think I put it, well, this shows your blood pressure. Here comes my incoming blood to, this is external respiration, so this is the lungs. My O2 is 100, my CO2 is 40. In my blood, my O2 is 40, my CO2 is 45. Where's my higher concentration of CO2? In my blood. Where's my higher concentration of O2? In the lungs. So we're going to switch. O2 to the blood, CO2 to the lungs. And when we come out of there, now my blood has high O2, low CO2. And then we're going to go to internal respiration. Here's my bloodstream. I'm now at 95. I've lost a little oxygen along the way before it gets to here. But now I'm at 95 on my O2, 40 on my CO2. Here's my tissues. I'm at 40 O2, definitely higher out here than here, right? So oxygen's going to go here. CO2 is at 45 versus 40. CO2 is going to go in. So we're just following the concentration gradients based on oxygen and CO2. All right, who transports O2 and CO2? Well, my red blood cells mostly do. Red blood cells transport my oxygen. They transport some of my CO2. Some of my CO2 floats in the blood, and some of my CO2 gets converted into a buffering system. And we'll talk about that. But for simplicity's sake, right now, we're going to see CO2 to the blood, red blood cells, O2 into the tissues. All right, so oxygen binds to the iron in my hemoglobin and forms oxyhemoglobin. Each red blood cell has about 280 million hemoglobin molecules. Every hemoglobin molecule has 4 iron binding sites. So each hemoglobin can carry 4 O2 molecules. So one red blood cell can carry a heck of a lot of oxygen, right? Is everybody good? Hemoglobin saturation, this just is talking about the percentage of hemis. What are my hemis? These guys, my iron binding spots in my hemoglobin are my hemis. We have 4 hemis per hemoglobin. So a hemoglobin saturation just means how many of those hemis, those iron binding sites, have oxygen in them at any given moment. What affects the amount of oxygen going into my red or attaching to my red blood cells? Partial pressure of oxygen. Blood pH. Remember when we talked about what a tight rein the body keeps on pH level? Why? Because acids denature proteins, right? So what's our range of pH? 7.352. 4.5. That is a tight pH range. Who's our number one thing that makes us go acidic? CO2 makes us go acidic. So if our blood pH level is getting too acidic, we are going to begin to put more oxygen in there. We're going to bring those oxygen saturation rates up. Temperature, we said higher temperature, higher saturation. And then metabolic activity within the red blood cells. The curves, I'm not going to quiz you on the curves. Just know that different factors, those factors we just talked about, will create a difference in the amount of oxygen saturation to our tissues. So basically, just for an example, I'm not going to quiz you on this, but when our pH drops or temperature rises, we're going to get more oxygen into those tissues in this case. So our tissues would receive that oxygen, which would change our saturation rates. So this just shows, it just is a curve that shows that. I'm not going to quiz you on those. This just shows different of those scenarios. With your red blood cells, they only generate ATP by glycolysis, which is that first step of the cell respiration cycle, it is anaerobic, right? We didn't need oxygen till the second step. So we can break glucose into our pyruvates without any oxygen present. That's what hemoglobin does. So it forms lactate, which forms also these BPGs. These affect basically our O2 binding and release rates. When our BPG levels rise, we're going to see our saturation rates change. When do they rise? When we increase pH or some of our hormones. All right, fetal hemoglobin. All you need to know about fetal hemoglobin is it has a higher affinity. It pulls oxygen to it harder. It has a higher saturation rate than maternal hemoglobin does, which makes it where mom will let go of her oxygen at the placenta and allow the baby blood cells to take it. We also talked about how they think that's what contributes a little bit to jaundice. As we break down those baby hemoglobin, those red blood cells, it creates a lot of bilirubin. And this just shows, you can see the fetal hemoglobin has a higher saturation, a higher affinity for oxygen than adults do. Carbon monoxide, it is a big problem. Why? Because it pulls to these iron binding sites harder and attaches easier and more firmly than oxygen does. So when someone gets in a exposure to carbon monoxide, it will slowly begin to fill those binding sites and it won't let go of them. So eventually you have all these red blood cells, but none of them can pick up any oxygen because the binding sites are full with carbon monoxide. And that's when people pass and die. They basically suffocate from lack of O2 because carbon monoxide is filling all the iron, the hemis, all the iron binding sites. CO2, how do we move CO2 in our blood? There's 3 ways we move CO2 into our blood. One, we can convert it to carbonic acid. This is how most of it moves, is right here. The second way is we can put it on the binding sites. So we can, well, into the red blood cells. We can release the oxygen, bring the carbon dioxide right into the hemoglobin of the red blood cells, or we can transport it as a dissolved gas in the blood. And we just saw the little picture where we can push gas molecules into liquids, right? So we can do that. We can push it right into the liquid and haul it into the liquid. This is how the least amount of it travels is as dissolved gas. All right, 70% of it goes as, basically we're gonna change it into bicarbonate ions. So we build that acid that we were just talking about. But if we just created all this acid and we dumped it into the bloodstream, what would happen to our blood? It would move way down here, right? And we're creating an acid with it. So not only do we create an acid, but your body creates a buffer with this, along with this acid. What's a buffer do? Resist changes in pH, right? So it takes these carbon dioxide molecules in, changes it into an acid and a buffer, and kicks it out into the bloodstream. Now the buffer is going to prevent us. We still do this slight shift to an acidic side. This is why CO2 causes us to go more acidic, but we stay within a normal range, or we pick up our respiration rate. So what happens? CO2 diffuses into my red blood cells. It gets converted to carbonic acid, which dissociates a hydrogen, which is going to make it acidic. But it also makes bicarbonate, which is a buffer. So it brings that in, it turns it into an acid, which dissociates the hydrogen, but it also kicks a buffer out there, which is going to prevent us from doing that big acid swing. It trades with chloride ions. We call it a chloride shift. All right, So that's the number one way that carbon dioxide is moved. We absorb it into the red blood cells, we turn it into an acid basically, and a buffer, and we kick it out into the plasma. We trade it with chloride. All right, CO2 can also be bound to our hemoglobin molecules. So just like we transport our oxygen, we can put it in here. That's only 23% of it. And then 7% of it can just be a dissolved gas. So it's the number one way we move CO2. We do the acid and the buffer. All right, so this shows here CO2 comes into the bloodstream. 23% binds to hemoglobin, 70% becomes carbonic acid. I'm sorry, it's a carbonic buffer and an acid. And this just shows that when we get to the lungs, we can trade it. I'm not going to walk through this whole thing just because here's our exchange with our chloride. When we get back to the lungs, basically what you need to know is the acid and the buffer go into the red blood cell, and we basically reverse the reaction. We kick chloride out, and now it's come back into carbon dioxide, and we can exchange it there. So we basically, when we get to the lungs, we just reverse that reaction. How are we doing on time? We are almost there. Let me see where we're at real quick. Oh, Vay, we're so close. How do you guys feel? You want me to finish it next time? We can finish it next time. So it won't take us very long, 5 or 10 minutes next time. We are then to the digestive system, which in my opinion, the digestive system is the easiest system that we do in CO2. Or in CO2. Oh my gosh, I've had such a hard time talking today. 202, my apologies. Do you guys have any questions about respiration so far? Okay. Awesome.
And we don't have any to do it for Thursday.